These webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website at autism.org. Now, before we get started, I'll introduce our speaker. Dr. James McPartland is the Harris Professor of Child Psychiatry and Psychology at the Yale Child Study Center. He is a neuroscientist and practicing child psychologist who directs the Yale Developmental Disabilities Clinic. Dr. McPartland is also a founder and director of the Yale Center for Brain and Mind Health and the Principal Investigator of the Autism Biomarkers Consortium for Clinical Trials, a US-based effort to identify biological indices to enhance intervention research in autism. Dr. McPartland's program of research investigates the brain basis of neurodevelopmental conditions to develop biologically based tools to improve clinical care and quality of life for autistic people and their families. And now I will turn this over to Dr. McPartland.
Thank you so much, Denise, and thanks everybody for joining. It's really my pleasure to have the chance to speak with you today about something that I think is very important, which is trying to see if we can harness science, specifically neuroscience, to improve the way that we care for and support autistic people. And so that's going to be what I'm going to talk about. These are, in full transparency, these are the groups that support my lab and me as an individual. I don't think there's any conflict of interest what I'm going to talk about today. And this is a broad overview of what I'd like to talk about. I want to really quickly touch upon the way that I conceptualize autism as a neuroscientist and how that makes it important but also challenging to study. I want to talk to you a little bit about what biomarkers are, how we define and operationalize them, what are some of the scientific and also practical considerations for applying them in autism, and then I'll walk you through an example, one that I've been very involved with, a particular index from the brain, a particular readout of the face processing system that is very, very relevant to understanding social communication in autism. And then I'll talk about some limitations on how far we can go with some of this work. I'll close by talking about some of the ways forward, different kinds of studies that we can do to get greater insight into how neuroscience can help clinical practice, like the Autism Biomarkers Consortium for Clinical Trials, which is such a mouthful that I will henceforth refer to it as the ABCCT. I'll talk about some work we're doing in the lab to increase the inclusivity of our research, and then some work that we're doing to see if this science can help us actually translate into therapies or supports for autistic people. So let's start really big picture. When I talk about autism, what am I talking about? So this is not something that I made up. These are the DSM-5 criteria for autism as I see them. It's a developmental condition. It's something that has to be present early in life, and that it affects a lot of different domains of function in a lot of different ways for different kinds of people. These include social and communicative ability. It can include things like what you're interested in and how flexible you are in your behavior or your interests. And then it can also include your sensory perception and response and your motor behavior. So this is a really, clearly these categories are broad and encompassing. There are a few things that are really important to keep in mind when we think about them in autism and how we think about autism itself. Right now, autism is something that we define entirely based on behavior. I'm going to talk to you about science today and biology, but it's really dissociated from the way we diagnose and operationalize autism, which is all on behavior. Behaviors that I can see as a clinician working with a child, and behaviors that I learn about from parents or caregivers or partners telling me about them. So really, right now, we rely on behavior. We know that there's more to autism than behavior. We know that autism is genetic. We know that it's based in the brain, but we also know that this is complicated. It's not like there is just one cause of autism. There's not just one gene. Even if we don't think about causes or etiology, when we think about all the different kinds of brain systems involved in autism, it's also very likely that there's many different brain mechanisms involved in accounting for the diversity of systems that are affected in autism. So we've got this behavioral definition, we've got this really complex biology that is incompletely understood, understood. And where that leaves us is in a place really where we don't have any biological assays that we can use for clinical purposes, not to diagnose, not to determine whether or what treatment is indicated, not to measure whether an intervention or support was useful in helping that person. And I see that as a significant shortcoming. I'll say it explicitly. It was in my introduction, but I really have two jobs here. I'm a clinician. I run a clinic that is primarily evaluative and diagnostic, but I'm also a scientist. And nothing that I do in my clinic really, nothing I do in my lab am I presently able to apply in the clinic. And if we could measure this kind of thing, if we had a biological marker, or the term that we use is biomarker, I think we could do so much better. And that's where I think we stand. We need these kinds of biomarkers to help us understand autism in a way that lets us do a better job. I'll just illustrate this to you. These are some of the scientific tools that I have at my disposal when I'm being a scientist in the lab. Lots of different ways of measuring brain activity. This is EEG, which I'll talk a lot today. This is positron emission tomography, functional near-infrared spectroscopy, eye tracking. So, all these really powerful, fancy tools that we use as scientists. And when I go into the clinic, the tool that I use is my eye, or the communication to me from the eyes of a parent. We literally are relying on the same technology that Leo Kanner did when he first wrote about autism in 1943. And that, in a nutshell, that's the work that we do in the lab. It's try to see if we can get some of these fancy tools to help us do the things that are most important, which are supporting autistic people. So when I use the term biomarker, which is a jargony term, and when it's an important term, and the FDA, our Food and Drug Administration, that regulatory agency in this country, takes it very seriously, and they've defined it. And this is their definition of a biomarker: a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. And so there are a lot of words there. If you think about what that means, really, to me, in a nutshell, it means it's something that is biological, that you can measure, that's relevant to someone, how someone is doing, whether that person is doing well or doing poorly, whether that person is being intervened with or not. But that's, a biomarker. It's really a very simple idea. It's just an objective measurement of something that is an output of biology. When we think of the FDA, they have different kinds of categories of biomarkers. And the way to think about these is the purpose of the biomarker. Like, what could it be useful for? And the FDA jargon for this, and which you may see in some of my slides, is context of use. When you see context of use, just think the purpose, the context in which this could be useful. Now, the most common way that people think about biomarkers is as a diagnostic biomarker. 90% of the time when I talk about my research and I say I'm studying biomarkers, people's presumption is that I'm looking for some kind of test that can tell me whether a person is autistic or not. And that is one way we could use a biomarker. I think it's a really tall order because of what I said before, is that there's so many different ways to end up with autism that it's not clear to me that we'll find a biomarker that could tell us whether a person kind of passes that threshold or not. But that's one way we could think about biomarkers. Another way that we could think about it, and these are the FDA's terms, is a susceptibility or a risk marker. I think many of us would prefer to avoid the word risk now in the context of autism. The idea of this is that it tells you that someone is more likely to develop autism than someone else. So this might be something like that we could understand earlier in life that tells us this person has a higher likelihood of ending up on the autism spectrum. Another kind of biomarker is what we call a pharmacodynamic or response biomarker. And this is telling us that something has changed in response to a treatment. Like if we had a medication that we thought would target a very particular brain system that's impacted in autism in a detrimental way for someone, this would be a biomarker, like maybe a brain image that would show us that that system is functioning differently. There are prognostic biomarkers that tell us about a person's developmental course. Is this a person who's likely to go on and have autism and be edified and productive? Or is this someone who may go on to have self-injurious behaviors and not be able to live life in an independent or fulfilling way? And then there's another kind of biomarker similar, but thinking about it in the kind of context of treatment. And that is predictive. And that's estimating how a particular person would be estimated to respond to a particular treatment. And then the last kind of biomarker, and this is the one that I really am going to be thinking and talking about today, would be a stratification biomarker. And what I mean by that, you could think about as subgrouping biomarker, for example. But the idea that if we have this incredibly heterogeneous group of autistic people, and that is the autism spectrum, it's tremendously diverse, is there something that we could measure that would help us to find them in subgroups in meaningful ways? Maybe some might benefit from a certain treatment, others may not. Maybe some are going to go on to great success and others are going to need more support. But this is, I think of this as the lowest hanging fruit for us in autism, is that there's many different ways we could use biomarkers. That's why I go through this list, because I think it's very important that you come away from this talk understanding that a biomarker is not just a diagnostic tool. It is something that can be applied in many different ways. And this is the way that I really think about applying it, is saying, is there, can we define autism spectrum in more specific ways? And I also think that's an excellent example of that first issue that I described, the tension between science and clinical insight, in that, as a clinician, me and my other, clinician colleagues in DSM-4, we thought we had subtypes. We had autism and Asperger's syndrome and PDD-NOS, and in DSM-5, those clinically based subtypes were deemed unreliable and went away. But there have to be meaningful subgroups within this very diverse spectrum, and that can only improve our ability to do clinical work. And that's the objective of the kind of work that I'm talking about today. I want to next talk a little bit about how we might think about evaluating whether a biomarker is useful or is working. And this is not from the FDA. These are my ideas. But one thing I think is really important is, even though I said the very lofty aspirational goal of having something as a diagnostic biomarker may not be attainable, we would expect a biomarker to hang together with diagnosis, right? That it should look different in autistic people versus non-autistic people if it's telling us something useful about autism. So that's one thing I think that we would probably want to see in a biomarker. We might also want to see, aside from just that categorical, autism, not autism, if we think in a more nuanced way about all the different kinds of things that can be impacted in autism, like social motivation or eye contact or speech or sensory response, there might be biomarkers for lots of different things. And then what we'd expect for those biomarkers is not just are they associated with autism or not, but do they relate to performance in those different domains? Does a biomarker for a specific process tell something about a person's functioning in that area? And then we would also, the other side of that coin is understanding how specific that information is. Does understanding, we should be as specific as we can about what a biomarker measures and what it doesn't measure. And I'll give you an example. If I were to have something that I thought was a biomarker for speech, but I didn't know its biology is actually more related to cognitive or intellectual ability, it would be tricky, right? I might look at associations between that biomarker and a person's speech, and I would see associations because speech is going to be tied so tightly to intellectual ability. But then, let's say I had a really effective treatment that changed speech, but didn't change intellectual ability. Well, that biomarker might be useless for measuring it, because it's measuring the treatment's effectiveness, because it's not really tied to speech, it's tied with intellectual ability. And so I guess we want to, in a nutshell, we want to understand what they hang together with and what they may not be measuring as well. We have when we think about a developmental condition like autism, it's also very important for us to think about how a biomarker could apply across the lifespan. Because when I say autism, I could be talking about a 3-year-old, a 30-year-old, a 60-year-old. And when we think about the difference between a 3-year-old and a 30-year-old, we really expect different things in terms of how the brain looks and works. And so we may not be able to apply the same kinds of brain indices in those in those different age groups. But that's something that we can measure and test and understand. But it's something it's very, very important for us to keep in mind. For these biomarkers, we want to understand how they relate to behavior. And let me tell you what I mean by that. There are some biomarkers you might measure where it doesn't matter. The person's experience of gathering that data is unimportant for the biomarker. Like, for example, if it was a blood draw to get genetic information, a child is highly distressed during the blood draw, a child enjoys the blood draw, the genetic information is going to be the same. However, for the kinds of things that I do, measuring brain activity, if a child is really uncomfortable or distressed during one of our scans, well, that's what I'm going to be measuring, not the things that I think are I'm trying to manipulate in that situation. So that's really important for us to know, is how the behavior during during the act of acquiring a biomarker can influence that biomarker measurement. The last thing I'll mention is that we might also think that a biomarker could tell us something about clinical status. We might want to see the biomarker change as a person gets better, or if a person got worse, we'd also want to see that change. That would be important to know about a biomarker. I want to make 2 important points about these kinds of things that I think are relevant to biomarkers, are that to be useful as a biomarker, any one biomarker needn't do all of these things. In fact, you might not want one to do all of these things. If one did all of these things, it might make it problematic for a particular purpose or context of use. And I'll give you an example. If the context of use or purpose that we saw was diagnosis, we would know that it's extremely important that it's sensitive to diagnostic status. That would be the thing. However, being sensitive to changes in clinical status could be problematic. If a person got better and they dipped below a threshold, or got worse and they came back over the threshold again, that would really complicate its use as a diagnostic biomarker. Conversely, if we had a biomarker that we wanted to use for response to see how a person was responding to a therapy or an intervention, well, then sensitivity to change in clinical status might be the most important thing to show us that the systems that we think are relevant to impact are being impacted. And so I've spent a few minutes talking about biomarkers, but I think that it's really important that it's appreciated that a biomarker can mean a lot of different things, and that it's a moving target. We evaluate the biomarker for its utility in a particular situation, and from situation to situation, the information that we would use to estimate it as useful may vary. So I also, as a clinician who would like to, my idea is that something that we discover in the lab could change the way we practice. I like to think about practical things. Like, one, it's a non-starter for a biomarker if we couldn't acquire it in a way that wasn't viable in this population. People who may have limited understanding of language, or who might have high kind of sensory responsiveness. So we need to have technologies that you can use in children, that you can use across the cognitive spectrum, that you can use in people with varying sensory responsiveness. We also want technologies that are economical, right? How useful a biomarker could be will be limited from a public health perspective by how widely it could be applied, and that's going to relate largely to cost. We also would want to think about accessibility. For how easy is this technology to come by, even irrespective of cost. If we had a machine that was very useful, or if we had a machine that was useful but could only be applied in an NIH-funded Autism Center of Excellence, this would be a representation of the number of people that it might be able to reach in those blue areas. Conversely, if we had a technology that could be administered in any hospital or existed in any hospital already, well, then this would be its reach. Now, this would clearly be of much greater public health value than this. And so that's something that I like to think about as well. So with these things in mind, I'll tell you a little about a particular biomarker technology that I use, a way of measuring brain activity that's called EEG. EEG is an abbreviation for electroencephalography. This is a way of measuring brain activity by measuring the electrical activity that is produced by brain cells when enough brain cells fire all at once, even though individually it's a minuscule amount of electricity. When they fire all together, that can sum up so that it can be detectable from the scalp. It's still very, very small, very, very low voltage, but it's detectable. This is the way we measure it. We use this is called a geodesic sensor net. This is an elastic mesh. Inside that elastic mesh are these small rubber pedestals. Inside each pedestal is a little sponge with an electrical sensor at the base of that sponge. And then what we do is we dip this whole elastic mesh, sensors and all, into salt water. So the sponges moisten and become conductive. We stretch it over a person's head like you see here. And then those sponges make contact with the scalp. And we then have an electrical bridge for those signals to pick up the neuronal activity from the scalp. Generally, there's two ways that we measure brain activity. When a person is just kind of sitting, idling, and that tells us just something about how the brain behaves just when a person is being. Or we can kind of perturb the environment, make things happen, like sights, like a face, or sounds, like a beep or a voice. And then we have the opportunity to see how the brain responds to those particular kinds of stimuli. That latter approach we would call ERP, or event-related potential, meaning that there's some kind of electrical potential caused or related to that event. EEG is a great technology to use in autism. It's very widely applicable. Really, all you have to do is tolerate wearing a damp, stretchy hat. You can be very young, you can be very old, you can have a very high level of cognitive ability or a very low level of cognitive ability. It's sensory friendly in that it's really just this kind of stretchy net compared to other kinds of technologies where you have to scrape the scalp, for example. And it's also pretty movement tolerant. If a person is wiggling around while we're doing this, it's fine as long as they don't wiggle around for portions of the recording. It's also a useful technology in terms of practicality. EEG is extremely cost effective. It's costly to buy an EEG system, but once we have one, really the cost is just a person's time and salt water and latex gloves, right? So it's very economical as far as brain imaging goes. It's also very accessible. When we think about it, EEG is already used at a population level for screening for seizures, for screening for hearing difficulties in newborns. And for that reason, there are already EEGs in most, if not all, hospitals in this country and around the world. And so it's also very, very accessible technology. And then the last reason why I think it's very useful in autism is that when we talk about all the different kinds of aspects of ability and function that can be impacted in autism, when we think about the diversity of autism, the one thing that we know that has to be true definitionally diagnostically, that there are differences in social communication. And this is a technology that's been applied very effectively to understand social communicative development, even off of the spectrum, just in neurotypical and non-autistic people. And so that gives us a good frame of reference to try to make insights about autism. This is an example of an ERP, an event-related potential. This is an example of one that's very relevant to social communication. Let me tell you what you're looking at. So this, on the y-axis here, labeled microvolts, is the amount of electricity that we're measuring produced by the brain. You can see it can be positive or negative, and the further up this line goes, the stronger the signal in the positive valence or in the negative valence. On the x-axis, I'm sorry, I may have called this x, this is the y-axis. On the x-axis, we can see time in milliseconds. And so One thing I want you to note is that this goes up to 700 milliseconds, which is 7/10 of a second. So this is when we use ERP, we're measuring things that happen very, very, very quickly. That's one of its strengths. And so what you're looking at here is how the brain responds to seeing a human face. This is something that happens over the right posterior scalp where these green electrodes are. And we see this negative spike at around 170 milliseconds. And the way we name these components, these ERP components, is just based on that. It's N, negative electrical activity, happening at 170 milliseconds, 170. So this is important because this is showing us that in less than 2/10 of a second, the brain is recognizing a face as a face. Not this is before this is mom's face, this is before this is a happy or sad face. This is just it's a face. So the very, very most basic stages of face perception, which is a really important social process. Now, this blue line is a group of neurotypical adolescents and adults. The very first study I did when I was a graduate student was to look and see. So many groups had said that people with autism have difficulty recognizing or processing faces. And so we thought, well, let's evaluate this index and see if we see a difference. And so this red line is autistic adolescents and adults. And what we found was that even though when you look at it, you think this difference in amplitude or strength of signal is the most obvious, what actually turned out to be the most important was the timing. And that when we saw that the autistic people were slower, they had what we would call a longer latency to their N170. And this is important, we think, because it means at this very first stage of this complex face processing series of neural events, we're already seeing a difference, meaning that it could compound over time and kind of give them a disadvantage, making sense of this information. So this was this first study and as I go through a series of studies that we've done, this was published in 2004, so now 20 years ago, a series of studies that we've done over the course of 20 years to understand whether this could be useful in autism, I'll refer back to that original list. And so referring back to that list, we learned here is that this is related to diagnostic status, that the N170 on average is different in autistic from non-autistic people. The next thing we wondered is in this study was, is it telling us anything about meaningful behavior? Like it's one thing to measure a brain response in a lab, but does it tie together with something that we think like might be socially important? And we thought, okay, well, this is a face processing system and face recognition is important. So let's measure how good a person is at face recognition. And we just used a standard psychological test. And we found that the autistic people had a lot more trouble. They made nearly twice as many mistakes with face recognition, despite being just as intelligent as the non-autistic group. And then we also saw that this was related to this N170 speed, that how fast their brain responded to faces was associated. And so this told us another important thing in terms of thinking about biomarkers, but that we're seeing that this N170 latency or speed actually correlates with something that we think might matter, a function, face recognition. The next question that we wondered about when we tried to make sense of these data is this telling us something that is really relevant to social communication specifically, or is this more just a basic sensory difference? Might autistic people respond more slowly to everything that they see, that everything that we would show them in an experiment? And so what we wanted to do here was find something that would show that there's not a difference, that autistic people their brains were responding just like non-autistic people. And so we took advantage of an autistic strength, which is letters. So many people with autism are gifted with letters or reading. We call that hyperlexia. Those that are average tend to be as good with letters as you'd predict based on their overall IQ. And so we had an idea, well, what if we look at the brain response to letters as an example of a system that is working well in these autistic people compared to the face processing system, which might be a source of difficulty? And it actually turns out that you can study this with the same component. So when we learn to read, we actually get an N170 coming from the other side of our brain, from the left hemisphere, that shows us that we can read that alphabet. Doesn't matter the alphabet, whether it's English or Chinese, for example. And we can test that by comparing it to an alphabet that no one can read, a made-up alphabet of pseudo letters, they're called, that we could use as an experimental control. And so that's what we did. We basically compared how autistic people's brains would respond to faces versus something that wasn't a face but was similarly visually complex, a house. And we compared how their brains would respond to letters versus something that was similar but not meaningful, a pseudo letter. And what we wanted to know was if what we thought this is telling us something really specific to the social systems in the brain, we would see differences here but not here. Whereas if it's a general problem with speed of processing, we would see differences in both places. So that's what we did. And we also thought, hey, you know what? If we're going to look at the same kind of thing, why don't we do it in a younger cohort so we can get a sense of the developmental relevance of this biomarker? And so when we looked at the social conditions comparing faces to houses, we saw the exact same things that we had seen in the older adolescents and adults. We saw that the autistic group had more trouble with face recognition, and we saw that their N170 response was slower in the right hemisphere. So this is important. It's important in science to show that you see the same finding repeatedly. We call that replication. And it was also important because it's extended. It said, hey, we're seeing the same thing, and it's not the same age group. So we're seeing this N170 difference in a younger cohort, so giving us information about the developmental relevance. When we looked at the non-social information, the letters, we had a really, really different picture. We saw a picture of kind of similarities. We saw that the autistic people and the non-autistic people both did great on our behavioral tests, which were of decoding words and decoding phonemes and reading words. And then we looked at their brain response to letters in purple here versus the made-up letters or pseudo letters in green. We saw two things. One, as shown in this chart, that everyone is showing the kind of specialization, the stronger response to the letters that tell us that their brain has specialized for reading these letters. We also saw that there weren't any differences in speed. It wasn't that the autistic people were any slower here. The only difference that we saw between these groups was that the autistic people tended to involve more the right half of the brain that is usually involved in faces. And that's the difference in lateralization that you can see on this chart. So this kind of aligned with what we thought was happening, that we're seeing that this N170 is different in autism in children and adults. We're seeing that it associates with this important social process, face recognition, but we're also seeing that it's not just generic information. It tells us nothing about how good they are at letters. It's really relevant to the social domain. The next question that we asked about this is, well, could there be anything about the way that we're measuring this that could cause us to question what we think we're measuring? And so this is work that was written up in our lab by Tamara Parker, who is a Ph.D. student at Yale. And as it turns out, there can be a number of reasons why a person's N170 might be faster or slower. And one of the things that we know from non-autistic research is that looking to the eyes gives you a faster N170. And one of the things we know from autistic eye tracking research is that people with autism tend to look less at the eyes. And so what might be happening here is maybe we're measuring their brain response to faces, and the autistic and non-autistic people are looking at faces differently. And the thing that we would conjecture could account for what's happening. If the autistic people were not looking at the eyes, well, maybe that's the reason their N170 is slower. And that would be important to know, but that would also mean we should be just measuring where they look instead of measuring their brain activity. And so we did an experiment where we controlled where they look. We used crosshairs to kind of set it up so that when a face came on screen, a person would be looking to the eyes or looking to their nose or looking to the mouth. And what we wanted to know is when we control where a person looks in this way, do the differences go away? If we force everybody to look at the face the same way, do the autistic and non-autistic people show the same brain response? And so that's what we did. And it's not what we saw. So this chart's a little busy. Let me describe it to you. So you can see here, brain responses. So this is the N170 latency in milliseconds. This is when people are looking to the eyes. This is when people are looking to the nose. And this is when people are looking to the mouth. And the non-autistic participants are shown in blue, and the autistic participants are shown in yellow. And what we found is if we hadn't even worried about this and we just looked at the experiment, we would see the same thing that we'd seen before, that overall the N170 is slower in autism. But when we look at it in this more refined way, we found that not only does looking at the eyes not make the N170 difference go away, that's what drives the difference, is that the non-autistic people show a much faster response to the eye when we encourage them to look at the eyes, whereas the autistic people don't. And so this indicated to us that even when we're really, really circumspect about the visual information a person is taking in, even when the same things are hitting the retina, the brain in autism is responding in a very different way. And so what we're seeing, this is not just some kind of artifact of behavior, and moreover, it's robust to even when we perturb behavior in this way. The last kind of experiment about this N170 that I want to tell you about is a very preliminary experiment because it's in such a small and opportunistic sample. And so this is work that we did in collaboration, led by two med students at Yale, Shash Kala and Max Rollison, who actually are now, one is already out of med school and one is heading into residency. But there was a colleague here at the Child Study Center, Pam Ventola, who was running a research program on a behavioral treatment called pivotal response treatment, which is a well-validated naturalistic intervention, basically to teach children how to play and to teach them core play skills and to see how that improves their social interest and social abilities. And she was looking at this in preschool children over 14 weeks. And we knew from her study and from a number of prior studies that kids tend to get better. And so what we did was basically see the kids for an EEG before and after treatment. And what we wanted to understand is whether, as they got better, their N170 would change. And it did. And so what you're seeing here on this chart, again, on the y-axis is their N170 latency. And you can see here before treatment and after treatment. The dotted line is these 7 children on average, and each solid line is an actual child's values. And you can see that on average, and for all the children except for one, they all got faster N170s as they got better. Now, I say this is preliminary because this is a very, very, very small sample. But what's interesting is that this is in the direction, if we think the N170 is giving us some kind of readout about some of the kinds of social or communicative difficulties experienced by someone with autism, and we see some improving in the study, we're seeing the N170 change along with it. So suggesting that it may provide information about clinical status. So just to recap, right, I have this list of things that are important for a biomarker, and we've looked at a bunch of them with the N170. We've seen that it's sensitive to diagnostic status, that it associates with face processing in a specific and not general way. It doesn't associate with letter processing. It's relevant in children, adolescents, and adults on the spectrum. It's robust even when people engage with the visual stimuli in distinct ways. It's sensitive, potentially, in a preliminary sense, sensitive to changes in clinical status. And then it also, because we're working with the EEG, meets some of these practical criteria. It's viable in this population, it's cost effective, and it's accessible. So if this marker has all these things going for it, well, why did I start out by saying we don't have any useful biological tools? And that's what I want to highlight now. I want to say this is the story I'm telling today because it's the one that I know the most about, most of the work that I've done in my lab. But there are other biomarkers that have strong evidence, eye tracking biomarkers, fMRI biomarkers. A truth for all of them is that despite a number of studies that show potentially meaningful information about autism, it's not perfect. We don't know why it works sometimes or why it works in terms of showing a difference or providing useful information and why it doesn't. Now, at the bottom of this slide is every study that I at least as of 2023 that has followed up on that initial paper I talked about from 2004. So lots of studies. And in fact, one of them, this Kang paper, is even a meta-analysis, which means that it took all the preceding studies, kind of put all their data together and analyzed them as a group and said, wow, this is actually true across all these studies. But it's not always true, and we don't know why. And there's a number of different reasons. It could just be that this is autism, that autism is heterogeneous. So maybe this would be heterogeneous also. It could be that there's problems with these studies. Some of them are small or lack statistical power, and so they could fail to detect results or give false results. Or it could be different choices that we make experimentally. When you're doing experiment, color faces, black and white faces, crosshair, no crosshair, maybe these account for the differences. Those are problems that we could solve. There are other limitations in getting any of these potential biomarkers to a place of utility, and one is knowing how stable it is in a person over time. Most studies only measure it in a person once, but it's very, very important to know that what I'm measuring today in an autistic child is telling me something that is true about them tomorrow or beyond. And we don't know that. We also don't have any kind of frame of reference. For many things that we do, like if we wanted to understand whether a person's head circumference was average or large or small, we could download growth charts from the CDC, but we don't have that for these brain-based biomarkers. And so in every one of these studies, the difference is being calculated by comparing autistic people to an idiosyncratic group of non-autistic people. And we could come to kind of norms, to understandings of what should be expected for an age group, for example. And so all of these kinds of problems could be solved with a different kind of study. And that is why now in 2000, now a decade ago, and the National Institutes of Health and others basically said, hey, what we want to do is get together a consortium of sites who will take the most promising biomarkers and study them in a way to really figure out what's what. Like, are there ones that we can cross off the list? Are there ones that we could pursue? And that is how the ABCCT came into being. This is a multi-site study, 5 sites around the country. It's not a clinical trial. It's just naturalistic, meaning we observe people over time. We chose the biomarkers from EEG and eye tracking, which had the strongest evidence. We clinically characterized people in very deep ways, so we have lots of information to understand how those biomarkers related to them. We had big samples. We studied them over time, so we could see reliability within a person over time. We used the reason we picked eye tracking and EEG, because these are practical, low cost, easily deployable technologies. And we also involved a blood draw so that down the line we could look at genes. The first phase of this study extended from 2015 to 2020 and included a large sample of 399 kids. Other things that I'll mention briefly that are special about the ABCCT, one is that it's not just run in academia. It's leadership. I lead a steering committee that involves people who work for the government at NIH, who are in academia, and then also we collaborate with experts in biomarker development in industry. So bringing lots of expertise to bear. We planned a very rigorous study. In terms of the way we run the study, we treat it as if it were a clinical trial and use the highest standard of kind of regulatory ethics called good clinical practice. Methodologically, we were extremely conscious of any sources of noise, making sure, for example, that the lighting levels in every lab around the sites around the country were all identical. And then statistically, we were very rigorous about pre-specifying our predictions and measuring them in reliable ways. The other thing that was unique is that we planned ahead and worked in tandem with a European consortium called, at the time it was called EU AIMS, now it's called AIMS to Trials, so that we could compare these results across studies eventually. I won't go through this list, but just to give you a sense of how comprehensively clinically thorough it was, these are all the different kinds of clinical measures that we involved in the study. And then we had 9 different biomarkers that we looked at. And the one I'll highlight is that one of them is the one that we've been talking about today, these ERPs to faces. Now, what did we learn from doing this different kind of study? Well, one thing we learned is that what we thought was true turned out to be true. We replicated things, which is really important. So in this very rigorous, large study, very heavily controlled with multiple sites, we saw the kinds of things that we'd seen in smaller studies in my lab and other labs, is that this N170 is slower in the autistic people. We also learned that when we plan this well, it's very effective in getting the data. We get data from almost all the non-autistic kids and from 3/4 of the autistic kids, which is actually, I'm glad to talk about it in the question and answer session, very high for this kind of research in children with neurodevelopmental conditions. We saw that it was pretty stable, both over 6 weeks and over 6 months. A person correlated with themselves, a measure that we call an interclass correlation coefficient, 0.66 in autism and 0.75 in typical development. And then we saw that we also saw, as we'd seen before, that this was associated with function, that those with faster N170s were better at face memory, and that that N170 today tells us something about face memory down the line. So this was important, and this was a study with a level of rigor that provided clarity that couldn't, that couldn't be drawn from the smaller preceding studies. And so we did something that hadn't been done before in autism. We approached the FDA, and the way you do this is you send a letter of intent to the biomarker qualification program of the FDA. You present your data, and you say, and basically you say how you think you're going to use this, what would the purpose of this biomarker be, and they say whether they think it's worth studying. And we did this for this N170. We did it for another eye tracking index called the ocular motor index, how much a person looks at faces. And both of these biomarkers were accepted into the FDA's biomarker qualification program, which is a milestone. This is the first time a biomarker has made it to this stage for autism. It's the first time that a biomarker made it to this stage for any psychiatric condition. So really, this was a big step. It's also a really challenging place to move forward. So what we said in our letter of intent is basically that we are going to use this as a stratification biomarker. Now, the FDA doesn't actually use the word stratification, so we said it's a diagnostic biomarker, but not diagnostic of autism, diagnostic of a subgroup. That this group of people who have slower N170s than almost everybody else are different from the others. They're going to be more biologically like one another, more biological homogeneous than the others. And then that could be useful information for planning clinical trials. We could analyze them separately. And that's what we've done. We've had, an ongoing series of grants to continue to work on this with the FDA. And we really now are in the process of wrangling through some really thorny, complicated scientific issues. How do we determine what's adequate in terms of being stable in a person over time? How do we draw a line in this distribution? How do we decide who's in the purple group and who's not? And then once we've decided who's there and who's not, how do we validate that with something else, with the clinical data or with other forms of brain imaging? Do we need a clinical trial to make certain inferences about how this stratification could be useful? And how generalizable are these findings? If we did this really rigorous study, do we have any concerns about whether this is something that would be useful in places that can be less standardized? We march on. The ABCC was renewed in 2020. We are now carrying out 3 studies in the second phase. A follow-up study, which is seeing that original cohort 2.5 to 4 years after enrollment. We concluded that study in August 2022. We're now in the midst of a replication study, seeing a different cohort of 400 children, doing the same battery that we did in the first study, so that we can do 2 things. One, replicate again, but then also have this combined sample then of 800 children, which will let us make much, much stronger inferences. And then we're now in the third study, which is a feasibility study to demonstrate to determine whether this battery could be useful in younger children, 3 to 5-year-olds. I want to close. So the ABCCT is unique in its rigor, but it's not doing anything. It's not looking at something that nobody's ever looked before. I want to tell you about 2 things we're doing in the lab to try and change the way we look at things and look at things that haven't been looked at before. This first line of research is led by Adam Naples in our group, and this is to address a big problem in autism neuroscience research, and that is the almost entire exclusion of individuals with severe, profound autism, those with low IQ. And what we've done here, I won't go into the details in the interest of time. I'm happy to go into the details in the question and answer session. But we basically built a special lab, a special experimental administration software, and used a very elegant behavioral approach that now is letting us reliably get data from people with autism who have much, much lower IQs than are included typically in studies like the ABCCT or other neuroscience studies. And it's working. I mean, so these are preliminary data showing that the N170 does what it should. We see a bigger response to faces than houses. We see, when we compare N170 latency, we see this graded effect that people with autism without intellectual disability are slower than non-autistic people, and those with autism and intellectual disability are even slower. And these data are from a group with an average IQ of 30, which is, extremely cognitively impacted. And so we're very encouraged by these findings and think this will give us an opportunity to be much more inclusive in autism research. The last thing that I'll mention is a different line of work led in the lab by Sherab Schleringa and Azieh Alsop. This is using brain stimulation to understand what these biomarkers are telling us and whether there could be therapeutic applications. Many different treatments that people in autism seek are designed to increase comfort in social interactions. We know that these are reflected in changes in a part of the brain called the superior temporal sulcus. And one of the things that we could do is directly stimulate that part of the brain to see, and some people have looked at this really, it changes social behavior. It can change kind of inflexibility of behavior. What we seek to do is pre-therapeutic, but really just understand the relationship between these biomarkers and this part of the brain. And what we've seen so far in our preliminary sample, these data are actually from non-autistic people, is that we see that it really changes some of these biomarkers in ways that are intuitive based on our understanding. That when you stimulate the brain, we see the N170 get faster. We see that that people look more to the eyes. And so we hope that this could be a way, should we determine that we can measure in some kind of objective way who stands to benefit or seeks to benefit from these kinds of therapies, we might have a way to actually change some of these things. So I'm going to stop there. I want to just express my gratitude to a few groups. Most importantly, the autistic kids, the autistic adults, and their families that go out of their way to spend time in our lab and help us learn together. The work that we do begins in our clinic, and these are some of the clinicians in the Developmental Disabilities Clinic at Yale who really do the most important job of serving the community and building trust and building relationships with those families who then are willing to get engaged with research. This is the broad group of people involved in the Autism Biomarkers Consortium, and then this is the group within the lab doing a lot of this work. Our email is up there. The hardest part of our job, believe it or not, is finding people who want to get involved in research. And it's hard, for example, in this Autism Biomarkers Consortium, we have sites in Los Angeles, we have sites in Boston, we have sites in Seattle. These are all really major metropolitan areas. And we in New Haven are trying to keep up. We want New Haven to be as powerful a force for learning about autism as any other place on earth. And it's hard when you're in a smaller community. And so if any of you are interested, or any of you know someone who might be interested in partnering with us in our research, please check out our website, email that McPlab address, and we would love to meet you and to work together. And I will stop there, and I'm happy to use our remaining time to answer any questions or address any comments that people may have had.
Great, thank you so much. We do have a few questions, so I'm going to go through these. One was an observation, so I'll share this with you. Got this over in the chat. This person says, we're doing biomarkers in the AIMS2 trials project. I like your premise of a changing biomarker. I've never thought of that. So sharing some information there. This person says, Dr. Elizabeth Torres has done some wonderful work regarding machine and biometrics to diagnose, characterize, and treat patterns of sensory motor control underlying autism symptoms. Guessing the two of you are familiar with each other. Just wanted to mention it since your work may be complementary.
Great.
Uh, this person's asking, can you elaborate on why sub-biomarkers such as Asperger's were taken out of the DSM or found unreliable?
Sure. So, and to be clear, this isn't work that I've done. I wasn't a part of the DSM working group that came to the definition. But the perception is that the diagnoses, the sub-diagnoses within, at the time it was called pervasive developmental disorders, weren't being administered reliably. That, for example, there was a study that indicated that as powerful a predictor of your language level, for example, of what diagnosis you would receive was what clinic you showed up at. That some clinics, for example, would be more likely to give a diagnosis of Asperger's syndrome than others. And that was deemed, again, not being in the room, that was deemed a problem in terms of reliability. The things that we want the diagnostic criteria that we put on paper to be the thing determining what diagnosis is administered, not, idiosyncratic local cultural differences in diagnostic practice. And so I think that had a lot to do with it.
Got it. Well, thanks for sharing that. We also had people asking about handouts from this webinar. I want to say the slides will be viewable in the playback. Dr. McPartland, if you want to share handouts, we can post them on the webpage afterwards.
Happy to. I'm happy to.
Yeah, that'd be great.
And that gives me a chance to redeem myself, because I think that there might have people in the lab that were on this last slide. So I'll fix that in the version I send out.
Okay, great. That's perfect. I'll just send you an email to remind you after the webinar. So we had a few other questions. This question is just a general educational question. So can you describe more specifically what sensory motor control means? What is that?
Sure. This is it's a topic much bigger than autism, but when we think about sensory motor control, we think about the circuit that involves sensing information in the environment and planning a motor response to it. We can also think about the sensation internally too of understanding your body's movement in space that helps you plan. And so that's something that is among many different spaces that is affected in autism. The work that I talked about today is, sensory motor is less germane to the work that I talked about today, really more sensation. That's what I kind of meant. It's just like, is there a difference in the way people see or hear, which could be related to, but is also could be independent from the way that information integrates with their motor system function.
This next question, actually, is next to her about the N170 topic. And the first one is, does the difference in amplitude in N170 response with the autistic individuals indicate a more subdued or challenged response? I think you addressed that, but maybe you can just go over that again.
So it's the two things. One is that, that across all these studies, that has been very inconsistent. In fact, in that first study where I showed the picture that probably encouraged you to ask that question, that wasn't actually, it didn't meet the criteria for statistical significance. So we don't really think about amplitude differences as being something that seems to be true across studies in autism. Aside from that, it would be very hard to infer from a difference in amplitude what it means for the experience of an autistic person, right? Because these are really basic, low-level kinds of responses. What we would infer is it could mean that fewer neurons are being activated. So, and what that means, maybe fewer neurons could mean a more subdued response. It could mean, less attention, less engagement. It's very hard to make inferences about the why. We can just see the what.
Okay, another question on this. Has N170 been compared to the intellectually disabled group without autism?
There are not many. It's a good question, and I don't know that complete literature offhand. There are the best way I can answer that question is saying, in most autism studies to date, there have not been strong relationships between IQ and N170 in the autistic or non-autistic participants. It's not like the smarter people have a faster N170 or things like that. It seems to track with something different, like face processing ability or perhaps kind of social communicative difficulty. That is why we think it's worth studying in those who are, severely impacted cognitively by autism, because it'll give us a chance to tease out kind of severe autism versus just kind of differences in cognitive ability.
We have time for a couple really quick ones. This first one, is it helpful to get EEG to attempt to understand any autistic child's brain activity? Would you feel like that would be helpful, or is there a specific time when you would feel that would be warranted?
Right now, all this stuff is experimental. If you have an autistic child, um, it's not presently going to give us useful clinical information to do an EEG on them, with the exception of someone worrying about seizures, in which case an EEG would be indicated to empirically test for the presence of abnormal electrical activity. I mean, I would love, give me another 20 years, and then hopefully we'll say, yes, come into the clinic, we're going to do an EEG, and it's going to help us guide our clinical practice, but we're not there yet.
Yeah, and it's a tricky procedure for a lot of people. It's hard even, with anyone. So yeah, definitely something to take into account. So last question. With the expertise that occupational therapy has in neurodevelopmental and sensory integration and autism, are OTs ever part of your studies?
Well, Brian, our studies really are really different from our clinical activities, right? So in the clinic, we have lots of different kinds of disciplines involved, but in our research studies, it's really mostly kind of neuroscientists. Um, in our lab, we have a number of clinicians involved, but their role is more kind of diagnosing and working with the families and giving recommendations and things like that. The science is less informed from a multidisciplinary perspective. That said, at Yale, there are very few OTs on the faculty in the psychiatry and child psychiatry department. It's a place where growth is needed.